Everybody? How are you guys hanging? You ready for the weekend? Hey, Dr. Judd. All right. It's 12:30, so we'll go ahead and get started. A couple of announcements. Lab next week is reserved for data collection. So it's really pretty cool out right now. So for example, if you're in the pollen load group, in one of the pollen load groups, we only have one pollen load group. If you are in the pollen load group, which is one of the Tuesday morning groups, it's going to be cold Tuesday morning. So what you want to do if you're in the pollen load group is you want to be collecting insects when it's nice and warm outside. And so my advice to you would be to not attend lab that day and to schedule a time either later on that same day or some other day next week. It's going to be getting warm during the daytime, but it's not going to be warm at 7:45. That reduces the activity of insects, and so the estimates of insects that you're going to get are going to be lower than normal, and you'll have a harder time getting the number of insects that you actually need. So if you're in one of the other groups, either the group that is gathering life history data on goldenrods or the group that is looking at the formation of galls on goldenrod, or the group that is looking at pollen competition, because those are all primarily plant-based things. They can be done at any time of the day, but since we have lab scheduled at 7:45 to 10:35, you might as well come to lab. I'll be out in the field with you, so I can circulate around. I can answer any questions that you have. I can help you with technique, et cetera, et cetera. So that's a good 3-hour chunk where you can get into the field and get something done. If you're in the pollen load group, my advice is do something else productive during that time. Wake up as if you're going to go to lab at 7:45, get other coursework done, and then find a time during that week when you might be doing something for a different class. Do that on Tuesday morning and then go into the fields when it's warm to collect insects. When you go out to collect insects, if you need me to go out and collect insects with you, just let me know when you want to go, and I'll tell you if I'm free or not, and I'll be happy to go out there and help you get started on the insect collecting things. Still finalizing protocols, but hopefully those should be up very soon. But I don't expect probably anybody to be doing any field collecting over the weekend. For one thing, we don't have the protocols out, and two, we've got time reserved for that next week. Dr. Judd, do you think they'll need much more than one lab period for data collection for most of these projects?
I suspect not, especially if you follow the instructions that we're going to put on the protocol about where to go to get the plants. I think that it's likely that the most difficult one is going to be the galls if the sample size is limiting. So you might need more time to hunt. But if you go out as a group and you all share the load, it should be okay.
Okay, all right. So that's kind of our...
But once you bring your assessments back, you'll need more time in the lab. So just the 3 hours for collecting data in the field doesn't represent all the data you're going to collect. You have to do more work in the lab.
Yeah, and so our goal is to kind of get you out into the field and get the field work done before it starts getting too cold. For some groups, if you're having to wait for seeds to mature and things like that, you may have to go out multiple times, but each of those times should not be terribly long after you've been out there once, because you're going to be marking plants that you're going to collect in the future, so they'll have orange flags on them, and you'll be able to go back and find those relatively easily to collect them in the future. So that is that. What other announcements do we need to make, Dr. Judd?
You guys have a week, I believe it is, before the final Hendry et al. Summaries are due. We should have comments back to you shortly. We're working on finishing those up as well as the protocols today, and we'll get those to you as soon as we can, so you have time to integrate all of that.
Yeah, my hope is to have comments on your results and discussion, or results and methods, back to you at the beginning of next week, so you have a week to have feedback in hand before you have to have the final version turned in. All right. I asked you guys to go and look something up for me. What was that?
The assumptions of Hardy-Weinberg equilibrium.
Okay, so the assumptions of Hardy-Weinberg equilibrium. Who was that?
Paula.
Oh, hey, Paula. Okay, good. So on the board behind me, I basically have what Hardy-Weinberg equilibrium says, which is basically that Hardy-Weinberg equilibrium says that allele frequencies will remain constant in a population, assuming that the following things are true. And one of them we already started talking about last time, and that was that there's no selection going on, no natural selection, no artificial selection, no sexual selection. So there are different forms of selection. Natural selection is selection imposed by kind of the environment in general. Artificial selection is selection that is imposed by human beings on natural populations, and or domesticated populations for that matter. And sexual selection is selection that is imposed by members of the opposite sex within a species. And so those are kind of three different types of selection. So one of the assumptions is that if there's no selection, then allele frequencies within a population should stay the same over time. What are some of the other assumptions for these allele frequencies to not change?
No mutation.
No mutation. So mutation brings about changes in the DNA. If the genes are determined by the DNA that makes up the gene, a mutation in the DNA will cause a mutation in the gene. And even though that mutation only occurs in one individual, that still changes the allele frequency slightly. So if you have 1,000 individuals in your population, because they're diploid, you have 2,000 alleles. If half of those alleles are the dominant allele, for example, and half of those alleles are the recessive allele, if there is a mutation that occurs in one of those individuals to a second allele, you still have 999 individuals with that allele, but you now have one individual with that allele. Well, that is a different allele frequency than what you had before that mutation occurred. So even though it may lead to small, tiny changes that are relatively rare, mutation does change allele frequency. What are other assumptions necessary for allele frequencies to not change?
Random mating.
Okay, so so random mating. When would you see non-random mating? Under what conditions would you see non-random mating? I've already mentioned it today.
In a lab setting.
What's that, Dominic?
In a lab setting.
Okay, well, normally we're thinking about this in natural populations. Out in nature, where would we not see random mating among individuals? So when we talk about non-random mating, random mating would be as if you have these genotypes in the population. This genotype is just as likely to mate with that genotype as it is to mate with other members of the same genotype as it is to mate with members of that genotype.
So,
And the same thing would be said of all of these things.
if I can just ask you to expand on that a little bit, why do you think it would be normal or natural in a real world field situation to see non-random mating?
That's part of how selection works, right? Is people or animals and creatures mate based on fitness and different features that are better designed to survive in the environment, and they attract people more likely than others.
Yeah, so that's Cameron? Okay. Yeah, so Cameron is exactly right. So we talked about sexual selection before. That's when members of one sex are selecting for members of another sex. This is where we get elaborate male traits and things like peacocks. Peacocks have really long tails because female peahens find those long tails sexy. They tend to mate with males more often that have those long tails. And over evolutionary time, this has become elaborated. And so random mating kind of falls partly under the no selection rule, because what random mating is saying is that there's no assortative mating, and that's what sexual selection is about, is when individuals mate with individuals because of their phenotype. In this case, are there other reasons why you might have non-random mating that aren't related to selection?
Maybe like availability. Like how many.
Expand on that. Who was that?
This is Tessa.
Oh, Tessa. What do you mean by that?
So like if there's a population that's smaller and there's not a lot of available mates, then their options are limited.
Okay, so that's getting there. Are you a member of a population now, Tessa?
Population, yes.
What population would you be a member of?
The human population, right?
Okay, but we've defined populations in the past. How do you define a biological population?
Oh, members of the same species in a relatively similar geographical location.
Okay, so what population would you describe yourself being a part of?
I'm part of like the population of Liberty.
Okay, you're a part of the population of Liberty. Do you interact with people in the greater city of Liberty more often than you interact with people on Jewel's campus, or do you interact with people at Jewel more often than people in Liberty?
People at Jewel.
Okay, so you might actually be able to define yourself as being part of an even smaller population, which is the population of people at William Jewell College. All right, raise your hand on the screen so I can see you. How many of you would consider yourself to be members of the William Jewell College population of humans? Okay, now, I'm not advocating this, but if you were going to mate with someone, are you more likely to mate with somebody on the campus of William Jewell College, or are you more likely to mate with somebody at KU, or at UMKC, or at Rockhurst University, or at Avila, or way out at K-State in Manhattan? Not that I'm advocating that you should be engaging in mating at all. But if you were, who are you more likely to mate with?
William Jewell.
Okay, you're much more likely to run into somebody that you might find attractive here at William Jewell. Why? Was that Alex? Who was that just spoke?
That was Connor.
Oh,
well,
Connor.
You'd see them more. So, I mean, I wouldn't even know who someone at KU would be.
Yeah, so if you never go to KU, you're never going to come into contact with that person. So another way in which you can get non-random mating, either among populations or within populations, although remember that Harding-Weinberg equilibrium talks about within populations, not among populations, but just spatial, non-random distribution of individuals in space can also lead to non-random patterns in mating. So even on the campus of William Jewell College, if you're in a fraternity and sorority, you might meet people in that circle of people that you wouldn't meet if you were not in the Greek system, for example. So you're all still members of the same population, but because you kind of run in different circles, you're less likely to mate with some people than you are perhaps with others. So non-random mating can come about because of sexual selection, but also can come about because of, sort of spatial dynamics within populations. Nathan, did you have anything to add to this?
No, that was great.
Okay, I'm cracking Nathan up evidently. What are some other things that are required for allele frequencies to not change?
Infinite population.
Okay, infinite population sizes. This is just another way of saying something else. What is another way of saying this?
Infinite resources and space.
No, this is infinite population size.
Oh, size.
If resources and things like that are constrained, what that's going to generally lead to is the presence of natural selection happening. Grant Latina, do you by any chance have a quarter, a nickel, or a penny or something in your pocket?
Maybe. Let me check real quick.
We got a penny.
Who's got a penny?
Joseph.
Hey, Joseph. Flip that penny, and we're going to play a little game. So here are 1, 2, 3, 4, 5, 6, 7, 8. Here are 8 genotypes on the board. When you flip that penny, if it comes up with a head, that means somebody dies. And if you come up with a tail, that means that they live. So for this first individual here, flip the coin and decide if they live or die.
It's heads.
It's heads. They died. So sad. Flip it again.
Tails.
They live. Flip it again.
Heads.
So sad. Flip it again.
Heads.
Oh, sad. Flip it again.
Tails.
Flip it again.
Tails.
Flip it again.
Tails.
And flip it again.
Heads.
Okay. So at the beginning, we started out with we started out with 8 individuals, which means we have 16 alleles. And so of these 16 alleles, at the beginning we had 1, 2, 3, 4, 5, 6, 7, 8. 8 of those alleles were the big A allele, 8 of those alleles were the little a allele. At the end, we have 1, 2, 3, 4 individuals standing. And unfortunately, that means there's 8 alleles, and there's 1, 2, 3, 4 alleles of each type. So the allele frequency hasn't changed at all, has it? This is the problem with Joseph's penny. It didn't work for us. Somebody else pull out a coin. Who else has a coin? Nothing against you, Joseph. It's just the way it turned out this time.
Grant has a coin.
Grant has a coin. Okay. Let me recreate this.
Here we go.
Wait.
Sorry, I'm just excited.
Oh, good. I'm glad that you're excited. Okay, so we still have 8 individuals. What's that, Nathan?
It likes the power to decide who lives and dies.
Yeah, it's cool. 1, 2, 3, 4, 5, 6, 7, 8 individuals. We still have 16 alleles total. 1, 2, 3, 4, 5, 6, 7, 8 of them are the big A, and 8 of them are the little a. Okay, Grant, heads or tails?
Heads.
Heads, dead. Heads or tails?
Tails.
Tails lives. Heads or tails?
Tails.
Tails lives. Heads or tails?
Tails.
Tails lives. Heads or tails?
Heads.
Heads or tails?
Tails.
Heads or tails?
Heads.
Heads or tails?
Tails.
Okay, so now we have 1, 2, 3, 4, 5 individuals surviving. That means that there are 10 alleles total, but there's 1, 2, 3, 4. 1, 2, 3, 4, 5, 6. 6 of those are the little a allele, which of course means that there must be only 4 of those. 1, 2, 3, 4 that are the big A allele. And so the frequency of alleles has gone from 1/2 in each of these to 2/5 and 3/5. So the allele frequency has changed. Did this change in allele frequency have anything to do with their genotypes?
No.
No, it didn't at all, did it? It just had to do with random death. These kinds of effects happen where the allele frequencies jump around because you have a small population size and a certain amount of random death just occurs. You're walking down a street and a piano falls on your head because they were moving a piano out of a tall apartment and the cable broke. You couldn't see it coming. You just happened to be randomly walking down the street and you were in the wrong place at the wrong time. You guys seen The World According to Garp? Oh, you should see the movie The World According to Garp. There is actually a scene in there where a piano falls on someone's head. It's sad. Anyway, random stuff happens sometimes. So that's what this is about. So when we talk about infinite population size, we're talking about getting a population size that's big enough that random death doesn't change the allele frequency at all. When the allele frequency changes because of these random changes, these random deaths, what's another term for that? What do we call that in evolutionary biology and genetics? Anybody know?
You might have seen it when you were looking up the assumptions.
You might have seen it when you looked up the assumptions. A three-word term, or sometimes a 2-word term. But I always put random at the beginning of it for me.
Is it gene flow?
It's not gene flow, but that's the next thing. So I'm going to just go ahead and put it up here. That's a different form of changing in allele frequencies. Who was that?
Joseph.
Joseph? Okay. So this process by which populations of small size change allele frequency randomly because of random mortality is called genetic drift. So another way of saying that there's an infinite population size is also saying that there's no random genetic drift. And some people refer to it as genetic drift. I always put the random on the front of it and say random genetic drift to just remind myself that it's a random process rather than any sort of directed process like selection would be. And random genetic drift is easy enough to illustrate just by throwing some alleles up on the board and then randomly killing them off with the flip of a coin. So the last of these is no migration. And so when I was a graduate student, I graduated, I went and taught at Shippensburg University for a year. And then while I was at Shippensburg, I applied for a postdoc in Puerto Rico. And Puerto Rico is a largely Hispanic territory of the United States. And when I got the job in Puerto Rico, I took all of my blonde-haired blue eyes. Yes, this is blonde hair. And I have blue eyes. And I took those alleles that are responsible for blonde hair and blue eyes, and they moved to Puerto Rico when I got a postdoc there. So even though it was just one individual moving, the pool of alleles that were largely non-blonde-haired blue-eyed now had the addition of a couple of blonde-haired blue-eyed alleles into the population. With that, I changed not only the allele frequency of the population that I was leaving from in Shippensburg, Pennsylvania, which has plenty of blonde-haired blue-eyed people, but I was then transferring those alleles to a place that was without those alleles in great abundance. And so that's another way in which allele frequencies can change, is just from individuals moving from one population to another. So these are 5, and I told you that we would only do 4, but I oftentimes think of non-random mating as being involved in sexual selection. So really it's no selection, no mutation, no genetic drift, and no migration are the ones that I want you to learn for the test. Keeping in mind that random mating is to a large degree due to sexual selection. Non-random mating is due to sexual selection. So I'll just redraw these on the board for you.
And for our purposes, when you hear non-random mating or assortative mating, we mean pretty much the same thing.
Yeah. Non-random mating, assortative mating. Yep, for the most part. When you get to genetics, we're going to give you something like 8 of these things. But for now, 4 is all you really need to know. And these are the big ones. The reason that I stick with 4 of these is that this also represents the 4 forces of evolutionary change. Evolutionary change can occur because selection occurs. Evolutionary change can occur because mutations occur. Evolution can happen because genetic drift occurs. And evolution can occur because of migration of individuals from one population to another. So these are oftentimes referred to as the 4 forces of evolutionary change. And in these, selection is generally viewed as reducing genetic variation. Mutation generally increases genetic variation. Genetic drift reduces genetic variation. And migration generally increases genetic variation. Selection is a little tricky because some forms of selection increase genetic variation and some forms of selection decrease genetic variation, which we'll talk about in a little while. But usually directional selection and stabilizing selection reduce genetic variation, whereas disruptive selection increases genetic variation. And we're going to be talking a lot about disruptive selection when we talk more and more about the finches in the Galapagos Islands. Any questions about this? All right, then I'm going to share my screen with you. Can everybody see the Hardy-Weinberg equilibrium slides?
Yes.
Okay, good. So here we are. We were talking about how we can take a single monohybrid cross and we can convert it into frequencies, the allele frequencies, and then from those allele frequencies, we can generate genotype frequencies. And so this is just one way of thinking about it. P is the frequency of the dominant allele, Q is the frequency of the recessive allele. P squared then is the frequency of two dominant alleles. 2PQ is the frequency of the heterozygotes, and Q squared is the frequency of the homozygous recessives. And this should always equal 1, which Dr. Judd pointed out to us last time. So here's a case with Manx cats. What do you guys know about Manx cats?
Nothing.
From what I can see
Nothing.
in the picture, they have no
Yeah.
tail.
Okay, so one of the characteristics of them, one of their phenotypic markers is that they have no tail. What else do you notice about that cat?
They're striped.
This one happens to be, but I think they probably vary quite a lot in coloration. So it has no tail. What do you notice about the body proportions of this cat? Does it look like a normal cat?
I think their legs are kind of short for their body and their body.
Yeah, the legs are kind of short for the body, and the body as a whole is kind of compressed from front to back. So these are kind of short, stocky cats. They're not cat-like in terms of their body form. And the other thing that they have is they have this lack of the tail. And so Manx cats are an interesting genetic case because the gene that converts a normal cat into a Manx cat is a dominant lethal. What we mean when we say that a gene is a dominant lethal is that if you are conceived in the early development of an individual, if you are homozygous dominant, you die during early development. You're spontaneously aborted, and you never make it out to see the light of day. And so if you are a heterozygote, you are a Manx cat, and you look like this little dude here. And if you're homozygous recessive, you're a normal cat. And so this is one of those situations where getting 2 doses of a gene has catastrophic consequences. Having only 1 dose of that gene just converts you into a slightly odd-looking cat. So the homozygous dominants are spontaneously aborted before birth. And this was first reported on the Isle of Man, which is one of the British Isles. And that's why they're called Manx cats, because people from the Isle of Man are sometimes referred to as Manxes. So there's a bicyclist who's known as Mark Cavendish is a bicyclist. He's a well-known sprinter in races like the Tour de France, and he is nicknamed the Manx Missile because he is from the Isle of Man. A little bit of bicycling trivia for you today on a Friday. So let's do a cross. If we have 2 Manx cats, and that's all the Manx cats that we have in our population, what is the allele frequency of the dominant allele for these 2 cats? If we have a population of 2 cats, 2 Manx cats. We have 2 cats. We have 2 alleles per cat. We have 4 alleles altogether.
What's the value?
How many of those?
P?
What's that?
What's the value of P in this case?
Yeah, what's the value of P? How many of these alleles are the big M allele?
0.75.
Not in this parental population.
2 or 50%.
Yeah, so 2/4 of them are the dominant allele and 2/4 of them are the recessive allele. Now, we let them breed on the Isle of Man. We get one individual that is homozygous dominant. We get two that are heterozygous, and we get one that is homozygous recessive. But what happens to one of these cats?
It dies.
It dies. And so when we get rid of that, P and Q go from what to what? In the ones that survive, in the cats that survive, what is now the allele frequency of both of these alleles?
1/4 to 3/4.
We have 3 individuals, so we have 6 alleles total. How many of those alleles are the big M allele?
Yeah, two.
We have 2/6, which are the big M allele, and we have how many for the little M allele? All you have to do is count them.
4.
It's going to be 4/6. This reduces to 1/3 and 2/3. So in one generation of breeding, we've gone from a population where P was 1/2 and Q was 1/2 to P being 1/3 and Q being 2/3 rather. So now if we want to see what happens to this population in the next generation, and I'm going to do this in fractions, even though I show decimals up here, because here in a minute, it's just going to be easier to work with fractions. So now we want this population to breed randomly. We want individuals to breed with one another just based on the proportion of individuals of each genotype in the population. How can we figure out what the genotype frequencies are going to be in the next generation if we know the allele frequencies? What can we use? What tool is available to us to use?
The p² plus 2pa equation.
You got it. We have 6 alleles total, 2 are dominant, 4 recessive, 2/3, or 1/3, 2/3. We can take those things and we can put them into an equation that will tell us what allele frequencies are. The Hardy-Weinberg equilibrium equation still works. It's just that it's not at equilibrium because clearly something's going on selectively. The thing that's going on selectively is there's selection against homozygous dominant individuals. So we can plug the 1/3 and the 2/3 into this equation, and what we get is we get 1/9, 4/9, and 4/9 for the different genotypes. So we would expect to see 1/9 of the population being homozygous dominant, 4/9 to be heterozygous, and 4/9 to be homozygous recessive. But of course, what happens to this 1/9 worth of individuals?
They die.
They do not make it out of the mother cat. So do we have 9 parts of our population? We only have 8 parts of our population, right? So that's at 8/8 is the 1 that E equals up to. And so this is really 4/8 and 4/8. So half of the population is made up of the heterozygotes and half the population is made up of the homozygous recessives. So now we can think about calculating allele frequencies as if we have a population of, once again, 2 cats that have these 2 allele frequencies. What's the, or 2 genotype frequencies rather. Half of them are this and half of them are this. How many alleles do we have total? How many alleles do we have total? Daniel, tell us.
Sorry. 4?
There are 4 alleles. How many of these alleles are the dominant allele?
Is it 2 or is it 1? It's 1.
It's
isn't
just
it?
one.
Yeah.
It's just that one right there, which means that the other 3/4 are the recessive allele. So now we've gone from a half and half mixture of alleles to 1/3 to 2/3 to 1/4 to 3/4. Now we can take that 1/4 and 3/4 and ask, well, what's the genotype frequency going to be in the next generation? Put in the 1/4 and 3/4 everywhere that they belong. We get 1/16, we get 6/16, and we get 9/16, adding up to 16/16. But what happens to this 1/16 of the homozygous dominance? Thank you, Grant. Can you speak that?
It dies.
It dies, it goes away. So do we have 16 parts of the population, or do we have another part of the population? We have 15 parts of the population. So we have 6/15 and 9/15. What does that reduce to? Reduces to 2/5 and 3/5 if we divide each of those by 5. No. Yeah, no, if we divide each of those by 3, rather. So we go from 6/15 if you divide the numerator and the denominator by 3, it reduces to 2/5 and 3/5. So that is the genotype frequencies. So again, we can think about this as a population made up of 5 individuals. 2 of those individuals are heterozygous, and 3 of those individuals are homozygous recessive. 3/5, 2/5 of them are this, 3/5 of them are this. So if we have 5 individuals, how many alleles do we have?
Ten.
We have 10 alleles. How many of those alleles are the dominant allele?
2.
We have 2/10, which is the dominant allele, which means we have 8/10, which is the recessive. That all equals out to 1/5 and 4/5. And so we have now moved from 1/4 to 1/5 and from 3/4 to 4/5. Sorry, I miswrote over here. 4/5. And so now these are the new allele frequencies that we have. So what's happening to the dominant allele over time?
It's being removed from the population.
It's becoming more and more rare. What's happening to the recessive allele? Somebody other than Cameron?
It's becoming more common.
It's becoming more and more common. Will we ever get rid of the dominant allele? Cameron is shaking his head no. Sophia, explain why.
Because it would still be there even if it's not.
Why is that?
Because it doesn't just go away.
So where is the dominant allele being harbored over time?
Wouldn't it still be like through the parent, but even if it goes down into recessive?
Well, the alleles aren't changing themselves, just the frequency of the alleles.
Would be,
Uh,
I don't know.
Go ahead.
I'm not sure.
Okay, call in somebody else in the class to help you out. We would do this actually normally in lecture also.
Katie, I don't know.
But Katie Sims has already answered something. Go ahead, Katie.
I actually do not know either.
Okay, you call in somebody to help you out. This is what it looks like. It gets closer and closer and closer to 0, but it never quite gets there. Why?
I'll call on Daniel.
Because since it's a dominant, it's going to stay, no matter what. So,
but why? What would we have to do if we wanted to get rid of the dominant allele altogether?
Eradicate the species, I guess.
No, we would only have to exert selection against 2 parts of the population. Really, we would only have to exert selection against 1 part of the population, because 1 part of the population is already having selection exerted against it. The homozygous dominants already don't survive development. What would humans have to do if we wanted to get rid of Manx cats?
You'd have to hunt down the heterozygous dominant and eradicate them.
We would have to hunt down the heterozygous individuals, and we would have to do something to get rid of them, because as long as you have heterozygotes surviving, you'll still have the dominant allele in the population. As a result, those heterozygotes get rarer and rarer over time, but it's very hard to get rid of them once they're in the population because the heterozygotes live. How many of you, raise your hands on camera? Let me see your faces and bodies. How many of you have ever seen a Manx cat in the flesh? Is it just Dr. Judnai? Oh, and Connor. Who else? Okay. Relatively few people have seen Manx cats. Well, this is the reason why. There's selection against the homozygous dominant carrying this gene, but there's not selection against the heterozygote. So you still have Manx cats that are very, very rare, but you don't ever really get rid of that particular allele because there's not similar levels of selection against the heterozygotes as there is against the homozygous dominants. Questions about the Manx cat thing? This is one way of applying the Hardy-Weinberg equation to simulate what happens in a population that is not at equilibrium because of selection in this case. As soon as you have selection acting on a population, equilibrium no longer occurs. The allele frequencies change. So this is a population that is not at Hardy-Weinberg equilibrium, but that doesn't mean that the equation doesn't still apply to it. It's just that the predictions of the equation is that the allele frequencies do change, because in this case, we're violating one of the assumptions of the Hardy-Weinberg equilibrium. Selection is occurring.
When I learned about this in an undergraduate class, an example we had in person, which we can't really do here, is if you imagine a bunch of red beads and blue beads, or red marbles and blue marbles, and you put them into a jar, and you start picking them out in pairs. Sometimes you get 2 reds, sometimes you get 2 blues, sometimes you get a red and a blue. If you're doing that randomly with respect to color, you've got a blindfold on, and you're putting those different colors, which represent alleles, together, you're going to get a proportion, a p squared, a 2pq, and a q squared of genotype frequencies. It's about at what Hardy-Weinberg equilibrium would predict, given how many reds and how many blues there are. But if you go in and you start looking, you know, I think it's really hot when all the red ones get together and make new red pairs, then you're violating that randomness, and you'll still get some total number of alleles and some total number of pairs or genotypes, but it won't be in the proportions predicted by the Hardy-Weinberg equilibrium equation.
Yeah, and we actually have bags of red and clear beads upstairs in White Science 224. If you want to go and demonstrate this to yourself, you can pull the beads out of the drawer and demonstrate this exact idea.
I'm sorry, I have another question.
Yeah, Tessa.
Okay, so with like Punnett squares and stuff, if we had like a heterozygous like parent and then like a homozygous recessive parent, then it's only like a 25% chance that one would carry the trait, right, and be heterozygous.
Yeah.
Is that right?
So let's just do the Punnett square. This individual can produce 1 type of gamete. This individual can produce 2 types of gametes. So we would end up with half of the population is heterozygous and half of the population is homozygous recessive. And so the allele frequencies here would still be P is 1/4 and Q is 3/4, and it was 1/4 and 3/4 here. But if we're not just thinking about individuals, so here's maybe something that will help you. If we're not thinking about individuals, if we're thinking about populations, and let's say that we have 1,000 individuals, and 3/5 of that population are homozygous recessive, and 2/5 of that population, well, let's just make it easier. Let's do 1/4 is heterozygous and 3/4 of the population are homozygous recessive. We're not just talking about 2 individuals mating, because this is 250 individual cats. This is 750 individual cats. Now, in many cases, what's going to happen, because we have 1,000 cats total, because 750 of them are homozygous, we're going to get a lot of breeding between heterozygotes and other homozygous individuals, homozygous recessive individuals, and other homozygous recessive individuals. We're going to get less breeding if it's random between homozygous recessive individuals and heterozygous individuals, but there will be some breeding between heterozygotes. And for those individuals that are heterozygous, it's going to produce these skewed representations of the dominant and recessive alleles. But to work this out in Punnett squares, what we would have to do is we would have to do a Punnett square of these 2 individuals weighted by the proportion of matings we would expect at random. We would have to do a Punnett square crossing a heterozygous and a homozygous recessive weighted by the frequency we would expect those to be if these individuals were mating on random. And then we would have the offspring from this population, from this cross rather, homozygous recessive crossed with homozygous recessive, once again, weighted by whatever the proportion of matings are that would be represented by this proportion of those individuals in the population. So that is way more complicated than actually just plugging it into the Hardy-Weinberg equilibrium.
Right.
Equation.
Right. So I guess my question was like, whenever you like, it's half the population of like heterozygous, right? That are heterozygous. And so like whenever the like ratio gets like smaller and smaller of like the heterozygous and their like phenotype. So are you saying that we can't look at it as one individual because like the likelihood of them having like one kitten and it not having any of the phenotype and being homozygous recessive is really unlikely?
No, it's likely enough. And the fact that it's likely is why the recessive allele increases in frequency over time. So let's just take the situation that you're talking about. If we have 1,000 cats, half of them are Manx cats and half of them are normal cats. Once again, we would expect a lot of breeding going on here among the normal cats breeding with normal cats, but we would also expect a good amount of breeding going on between normal cats and Manx cats, and we would expect the same amount of breeding that's going on here to also be going on here, Manx cats with Manx cats. These individuals would all produce nothing but normal cats. These individuals would produce a 50, so this cross here would produce 100% normal cats. This cross here would produce a 50-50 mix of Manx cats and normal cats. We just did that cross. But this cross here would produce a 2/3 Manx cats and 1/3 normal cats. Okay? And so in 1,000 cats, randomly, how many of these matings would we get that would give us 100% normal? How many of these matings would we expect that would give us this 50-50 mix? And how many of these matings would we expect that will give us a 2/3, 1/3 mix? Because those actually should all be even, we're going to get more normal cats, then we get Manx cats, because we'll get 100% from this, 50% from this, and 1/3 from this, whereas we only get 50% and 2/3 from this, and we don't get any here. So that dominant allele becomes more and more rare, the recessive allele becomes more and more common. Does that help?
Yeah, I'm caught up now. Thank you.
Okay, all right, good. The reason that I don't teach it this way is actually because in my own brain, it's just more comfortable for me to use the Hardy-Weinberg equation than to try and figure out, well, what's the proportion of all these different types of crosses? And that's actually one of the great utilities of the Hardy-Weinberg equation, is that you don't have to think about it that way. All right, other questions? As you're having difficulty, anybody in class, this is not directed at Tessa, if you're not understanding something, if you ask us about it, we're going to do our best to explain it to you in a different way. So we know how we've explained it to you in class, and since how we explained it in class isn't working for you for whatever reason, we might try that attack again, and if you're still puzzled by it, then we're going to shift to a different way of thinking about it. And we're going to try out as many ways of thinking about it as we can until something clicks in you that says, okay, now I get it. And so this is an example of that. There are various ways of thinking about this. I usually go with what to me is the simplest one. Simply because it's a simple one for me doesn't mean that that's the one that's gonna resonate with you. So don't be afraid to ask about these things. All right. So the dominant allele is harbored in the population of heterozygotes, so you would need to have selection against the homozygotes and the heterozygotes in order to totally get rid of the population of Manx cats. But the flip side of that is Manx cats are very rare, which is why only 3 of us in the class have ever seen them. Dr. Judd and I have been alive for a longer period of time, so we've had more opportunities to see them, but they are still just very rare. So when we look at these types of traits that are not single gene traits, these multi-gene traits like this, that have these normal trait distributions like this, we can think about the different types of selection as leading to changes in the shapes of these distributions. So let's say that we have the phenotype that is height or plant height or human height or whatever. We have very few individuals that are very, very short, very few individuals that are very, very tall, and a bunch of individuals here in the middle. Well, the way directional selection works is it selects against one extreme or the other. It doesn't really matter. In this case, we're selecting against short individuals. What that has the effect of doing is it has the effect of moving the population, the next generation, to the right, away from the direction that selection is pushing it. So we select against this end, it shifts away from that selection. And so we see that there's a change in the mean, and that actually shouldn't be labeled S, that should actually be labeled R. I need to change these slides. R is the evolutionary response to selection. Now, I know that we've already taught you a big R beforehand about population growth, but this is an R about evolution. R is the evolutionary response to selection.
So make sure that you know which R you're thinking about when you're dealing with a problem, because we're going to go back to the other R in a couple days, a couple lectures.
On Monday, I think. Yeah. So keep straight in your head when you're dealing with R in a population growth context and when you're dealing with R in an evolution context. So there's a different type of selection called stabilizing selection. In this type of selection, selection acts against both extremes, but doesn't act against individuals of intermediate phenotype. What happens in this case is the response to selection doesn't change at all. Oh, that thing just went away. But what has happened is even though the average phenotype stays the same, so it's the same before selection as it was after the selection, what's happening in this case is the amount of variation in the population is going down. So this post-selection distribution is narrower than the pre-selection distribution. In directional selection, the post-selection population is also narrower in terms of variation than the pre-selection population. So in directional selection, one of the ways of thinking about different types of selection is that they are different in terms of what happens to the average phenotype and what happens to phenotypic variance. And in directional selection, the average phenotype shifts, and it can either shift up or down. It doesn't really matter. It depends on the exact form of the selection. So it shifts. It can either increase or decrease, but the thing that happens to the variation is the variation decreases. In stabilizing selection, the average phenotype does not shift, but variation still decreases. So this is directional selection. If we selected against tall individuals, it would shift to the left rather than shifting to the right. But in any case, the mean, the average phenotype shifts, and the variation in the population goes down. In stabilizing, there's selection against both extremes. In this case, the average phenotype does not shift, but once again, the variation goes down. The solid distribution has less variation than the dotted distribution. A third type of selection is disruptive selection. Anybody want to guess what disruptive selection is about? This is the type of selection that we're actually studying in the Galapagos Islands with finches. In the finches, who gets selected against?
The average, like the mode?
Yep, exactly. The most common, or the intermediate phenotypes are the ones that get selected against, because there are fewer intermediate seeds, there are more large seeds and small seeds, and so those individuals that can handle small and large seeds survive, and the intermediate beaked finches do not. What happens here is, once again, the average stays the same, but because now more of the density of the distribution is distributed away from the average value, we actually see the variance increasing. And so the total variation in this population that is undergoing disruptive selection is increasing, not because the absolute values of the shortest and tallest individuals have changed. It's just the distribution of where individuals in this population lie. They don't lie centered right over the average. They lie in modes that are centered a little away from the average, and that increases the total variation. So 3 modes of selection: directional, stabilizing, disruptive. You can quantify them, or you can describe them in terms of the effect of that selection on the average value, and also the effect of that selection on the variation. So when we're thinking about these types of traits, the way we measure heritability in quantitative or additive traits is actually by using statistics. And the way we typically do this, the simplest way of doing this, is to look at the average value of parents and look at the average value of their offspring. Whatever aspects of the phenotype that get passed on to their offspring are going to be evident in this regression line. So these are actually data from a Bio 133 class that we had a number of years ago. I asked them to go home and get the average, or call their parents and get the average heights of their parents and get the heights of all of their adult children. So assuming that your siblings have all reached their final height, we didn't want, if you had a 5-year-old sibling, we didn't want their height because they're not at full height yet. And what we did was we got those data and we just threw them into a regression model. And when you regress average offspring height, in this case in inches, against mid-parent height, the slope of this line represents heritability. Heritability the shorthand for heritability is h squared, but this is just a symbol. You don't have to do a square root of anything. And in this case, heritability is 0.9165. And heritability, in theory, has limits on it. Heritability can vary from 0 to 1. 0 would be a flat line, such that the average phenotype of the parents doesn't have any effect on the average phenotype of the offspring. And a heritability of 1 would be a slope of a 45-degree line, where the average size of the parents is exactly the average size of the offspring. Average size of the parents is exactly the average size of the offspring. And so heritability generally lies between 1 as a maximum and 0 as a minimum. Well, the cool thing about thinking about this relationship is that we can take this relationship and we can say, well, what happens if we prevent one portion of this population from breeding? The average value of this population before selection was, I don't know, what's that, 67 something. And that would have produced offspring of a particular height. But after selection, the population of parents is now taller than it was, such that they should produce a taller average offspring. This is the strength of selection. This is the evolutionary response to selection. It can be notated delta Z, or it can also be notated R. And the thing that determines how well selection translates into an evolutionary response to selection is heritability. So since we're out of time, I'm just going to give you this equation. The evolutionary response to selection is equal to the heritability times the strength and direction of selection. And that all comes out of this graph of how we measure heritability. And I'll need, I don't know, like 5 minutes on Monday, Nathan, to finish this off, I think.
Yep, not a problem. Just a reminder, when we switch gears on Monday to a different topic, Chapter 11, Section 1 and Chapter 11, Section 2 listed in the schedule are a good kind of preview of what we're going to talk about.
All right, any questions on this before we leave?
Can you say that equation one more time?
Yeah, the evolutionary response to selection is equal to the heritability multiplied times the strength and direction of selection. So selection can occur in a positive way where the mean is going up, or it can occur in a negative way where the mean is going down from generation to generation. So it's how strong is selection, and also is it a positive selection or a negative selection. Multiplied times heritability, that will tell you how much the population is going to respond generation after generation. And we'll actually work an example on Monday. All right. All right, then we'll see you guys on Monday. Hey, Nathan.
Nope.
What was so funny? You were cracking up early on in the class.
Oh, I was laughing at the laughter that I saw among the students about your example.
Okay.
It was working well.
Okay. All right. Well, good. Good. If we can't be anything else, we might as well be entertaining.
Right.
All right. Catch you later.
All right. See ya.